What is PrimeXtract? PrimeXtract is a rapid, user-friendly spin column extraction kit for the isolation of total nucleic acids from cells, bacteria, and viruses. The column and buffers are specifically designed to maximize RNA and DNA recovery. When used with specimens collected in PrimeStore MTM, the extraction yield is superior to other extraction columns available on the market. Why is PrimeXtract better than the competition? PrimeXtract spin columns ensure complete DNA and RNA elution with no lysis or wash buffer carryover. This results in contaminant-free, high-purity RNA and DNA for downstream PCR or sequencing. In contrast to other columns, PrimeXtract columns contain a tapered terminus, no O-ring, and a distal silica exit. Suitable specimens for use with the PrimeXtract kit include primary specimens or cultured samples, veterinary samples, environmental samples, or samples collected in PrimeStore MTM, including sputum, throat swabs, and nasal washings. PrimeXtract is completely compatible with PrimeStore MTM. PrimeStore MTM, in contrast to RNA later or viral transport media, is a sample collection solution that safely inactivates microbes and then stabilizes and preserves the RNA and DNA from those samples for downstream nucleic acid testing and next-generation sequencing. Most commercial spin column extraction kits contain a rounded bottom and an O-ring. What we found is that this increases contamination and affects the downstream purity of the extracted nucleic acids. In contrast, our kit has been re-engineered such that it has no O-ring, has a tapered bottom, and the silica is positioned at the distal end to increase extraction efficiency. Purification of nucleic acids using PrimeXtract is so simple a high school student can do it. The first step in the PrimeXtract procedure is to lyse the sample and bind the nucleic acids to the silica column. This is accomplished by combining lysis buffer with sample and ethanol in equal parts. In this demonstration, our high school researcher is adding combined sample, lysis buffer, and ethanol to the column and centrifuging to bind the nucleic acids to the filter. In step 2, the bound nucleic acids are washed by passing wash 1 solution through the column and performing another centrifugation. In our student demonstration, the column containing bound DNA and RNA is washed by adding 200 microliters of wash 1 solution and briefly centrifuging. After discarding the wash 1 eluate, a second wash, wash number 2, is added through the column, centrifuged, and subsequently discarded. After wash 2 is centrifuged through the column, the eluent is discarded and a second spin is performed. The column is then transferred to a clean microcentrifuge tube in preparation for elution of nucleic acids. The final step of the PrimeXtract procedure involves elution of purified nucleic acids from the column. This is accomplished by passage of preheated elution solution. Preheated elution solution is centrifuged through the column. The eluent contains the purified nucleic acids. Samples are first collected in PrimeStore MTM and then shipped at ambient temperature to regional or diagnostic laboratories where they're subjected to PrimeXtract and then amplified using PrimeMix and real-time PCR. Visit our website or contact us directly at Longhorn Vaccines and Diagnostics for information on our Prime Suite collection to detection product line that includes PrimeXtract, PrimeMix for real-time PCR detection, PrimeSeq for whole gene characterization using next-generation sequencing, and PrimeStore for safe sample collection and ambient temperature shipment. For orders or inquiries, contact us at www.lhnvd.com or call us directly at 210-826-0910.